Today I'm going to speak about germline versus somatic variation, and I think this is important because whenever we talk about tumor sequencing, we have to remember cancer is a disease of the genome, and so we began with our germline genome, what we were born with, or our constitutional genome, and over time there's the acquisition of somatic mutations, and this can lead to cancer, and so it's very important to consider that distinction when we're talking. Here you have 20 individuals, they all have cancer, and this is just sort of unselected cancer, say it's advanced cancer diagnoses. We expect one of those individuals to have an inherited predisposition to cancer. And so if you consider that a normal cell can go on to acquire mutations, and this usually happens over our lifetime, and it's usually, the main reason is just because our cells have to divide, and we've heard 3 billion pieces of information that need to be, that make up our DNA or our genome. These are the base pairs, and those have to be replicated each time the cell divides. So we're bound to make mistakes, and we correct them well most of the time, but if there's a problem or a mutation develops in a gene that's, say, responsible for DNA repair, then there can be problems with regard to correction, and you can acquire mutations. So over time, we realized that within a tumor, you get different branches of the mutations that develop, and so there's this idea of the clonal heterogeneity, which we've heard about, and that brings up the point, it makes a difference where you're actually sampling the tumor in terms of what mutations you see. So when we talk about sporadic cancer, how does that differ from hereditary cancer? Well, I would argue the quite simple distinction is in hereditary cancer, basically you're born with that first mutation. That's the initial molecular lesion that's relevant for why the cancer developed. That actually makes it a lot easier when you talk about things like tumor heterogeneity, and the mutations being low frequency, because we actually know that these occurred early on. In fact, these were the first lesions that were important. So it's basically in every cell of the tumor going forward. And we know that when we actually study these tumors, that the germline mutation that's involved in cancer susceptibility is retained in about 90% of the time. Okay, over time you do get a lot of changes, and so you can lose it, say as one of the resistance mechanisms, but more than less it's retained. So why is that, when we think about cancer susceptibility in general, and when we think about if you, me, anyone in this room were to sequence their genome, so take a blood sample, sequence it, we'll find genetic variation, and there's different estimates of what it is, and just say we have 4 million bits of variation each. And that can be rare variation, so things that are private to you or private to your family, or variation that's more common in the population. And so when we talk about why an individual would see the hereditary cancer program, we're interested in the rare variation that predisposes to a high risk of cancer. And so it's on the left-hand side of the curve. There we go. So just on that back screen there, that's the one that's useful for most people. So here, these ones here, the genes that were primarily found by linkage analysis. So these were what we call sort of informative families. So there are multiple cases of cancer that occurred in them, and we were able to study those affected individuals in order to find the genomic regions that were shared amongst the individuals. And then by studies of actually going down to and looking at the genes within those, identifying the mutation that was common to the affected individuals. And that was using mainly sort of other genomic technology at that time, because these were identified, in a lot of them in the 90s. We're identifying more and more each day with the new sequencing technologies that are available. I'm just going to point us over to the right side of the curve, because this is, we all carry variation, and 23andMe also characterizes our variation for the low cost of around $200, I think. And so in this respect, we can get an idea of disease risks, but it's not something that we generally can act upon. And so we're really focused on identifying the more private variation over here that predisposes to a high risk of disease. Where we are getting into trouble these days is actually these genes in the middle here, where you see the population frequency is a little greater, and the phenotypic effect size, so basically the risk of developing cancer with them, is actually not as high as these ones. And so it gives us difficulty in the hereditary cancer program in terms of how we use this information in terms of our cancer risk assessment. Up here, we can, quite confidently say that there are management strategies, and if we do targeted mutation testing within a family, those that have the mutation can undertake certain cancer risk reduction management guidelines, whereas those who don't carry the mutation generally go back to population risk unless there's something coming down from the other side of the family. Whereas in this case, we run into the difficulty because if somebody does not carry the mutation, are they truly not at risk of developing developing cancers in the family? So when we counsel patients, we counsel them that, in general, there's sporadic cancer, which accounts for about 70 to 75%, familial cancer, where we have that combination of the more common genetic variation probably and environmental factors that impact on cancer risk, and where we can't really pinpoint what that genetic risk is. And then we have hereditary cancer, where we can pinpoint those genes, and that was in that upper left-hand corner I showed you. And we probably know about 140 or so of those genes, and we're learning more every day. Okay, here again is just to emphasize the point that over time, this is in sporadic cancer, it takes a lifetime to acquire these mutations, basically, and generally it's sort of lightning has to strike twice for this 2 hits. So you have 2 copies of every gene, one from mom, one from dad. In the idea of the tumor suppressor, your cell is okay if 1 copy is mutated, but if something happens to that other copy, then this can lead to the acquisition of more mutations in cancer. The problem in hereditary cancer is that you're born with that first hit, and so the time to acquisition of the second hit is sooner. And so people are predisposed to multiple primary cancers, earlier onset cancers, and usually they're of a certain pattern. So when we talk about tumor normal sequencing, so before we were just talking about normal sequencing, now we're talking about tumor normal. What happens? So this is when you do the exercise where you actually sequence unselected cancer cases. So before I was describing sequencing the normal people highly selected, but when you just go and sequence them, and this is for the reason of looking for sort of somatics, so acquired mutations that are targetable, which was the main idea. In that case, germline blood was sequenced as well in order to actually subtract out the variants that were germline so you could focus on those acquired ones. And so this is a survey of what happened when we looked at this at Sloan Kettering in over 1,500 cases. They were mainly breast and lung, colon and prostate, and some esophagogastric adenocarcinoma, although there was a wide array in over 68 tumor types in total, all advanced cancer diagnoses. So when this panel basically came about because the scientists and clinicians were interested in their favorite genes, and as Stephen alluded to, these come out of more comprehensive studies of associations, and basically the idea was to make a panel that could rapidly sequence these, and it was an enterprise effort and to do this within the institution. And so I remember first initially looking at this panel and thinking, God, there's actually a lot of Mendelian disease genes on there, and I'm actually a general geneticist, and so seeing genes that I know predisposed to, say, mental retardation, or not predisposed, but are seen associated with it, or other totally not what I would consider cancer-related genes, I thought this was an important exercise that we actually needed to survey what the chance was of actually finding this information, and even on tumor panels, because we talk about worrying about incidental findings in whole genome panels, but even if you consider tumor panels, and it makes a lot of sense that a lot of the genes that are important in carcinogenesis are also the ones that were really important in embryogenesis. So when we did this survey and looked at which genes were actually associated with a single gene disorder, Mendelian disease gene, over 55% of the panel had associations. What it also showed was that 93 genes on there, so 27% of the panel, had a germline association. And this was partially, deliberate in order to try and make the germline panel overlap with the somatic panel. What's important, I say they're the ACMG genes, and some people may or may not know, but the American College of Medical Genetics and Genomics released guidelines to basically say that anytime any sort of clinical sequencing was undertaken, there should be an examination of genes, and so this is germline sequencing, examination of genes that have been associated with high penetrant, so high risk of developing disease genes, of clinical significance that would mandate sort of medical interventions. And so these are things like, in some cases, cancer susceptibility, which was about 26 of the genes or so out of the 56 genes that they recommended. Also, ones related to arrhythmia, malignant hyperthermia, and other such medically relevant ones. So what we found was that there were presumed pathogenic germline variants, so this was studying the blood in about 16% of the cases, and so this was Mendelian disease genes. These were all different types, so it was autosomal dominant, autosomal recessive, X-linked, X-linked dominant. And when you looked at the cancer predisposition ones, it was about 13%. Okay, and so this is very interesting. It was very interesting to me, who knows that our highly selected criteria for the hereditary cancer program has, a similar detection rate. And so what we also see here is that the ACMG genes, so these were the ones that had implications that were actionable, were seen in 6.4%. And I should say that the monoallelic mutation carriers are tied into that number, so it was actually about 5%. Okay, variants of uncertain significance were seen in almost all the samples. So that in itself is, it's important to see that because we talk about genome, but when we just consider panels that are smaller, and these were panels that were targeting the protein coding regions as well, so it wasn't even the full genes. Okay, so potential to expand known phenotypes. So when we looked at the BRCA1 and 2 mutated cohort, germline mutated, there were actually a lot of cancers on there that we wouldn't have typically have tested for, and that we wouldn't necessarily have said, there's a high likelihood of having an underlying mutation here. And the ones that came out of there were the gastric cancers and the sarcomas. All right, so potential to expand known phenotypes. And you can't see this, but there is a publication on this, so you can look in more detail if you'd like. But it shows that, you can see BRCA1 and 2 mutations in lung cancer. This is important. This is important because we have an oncopanel that has BRCA1 and 2 on it. It's important, what else is important is that we know that from other studies, if you see a BRCA1/2 mutation, there's about a 60% likelihood that it's germline, and that's different from if you see a p53 mutation on tumor sequencing when there hasn't been a subtraction of germline DNA variants. Okay, so now this brings up the recognition that there's actually germline variants in there, and those potentially have target or rationalized therapies involved in them. And Stephen alluded to one of those, which is actually the platinum-based therapies and PARP inhibitors in BRCA1 and 2. So what might we reveal from this is the exact prevalence rates will depend on the population characteristics, the variant curation of how we determine pathogenic variants, and our estimates of cancer susceptibility in that are around 13% in unselected individuals, 5% for the actionable ones. So that's important when you're thinking about consenting. And Martin alluded to the importance of pipelines is because you need quick resources and able to rapidly curate these variants. And you need pipelines in order to do that. So the mutations weren't confined to cancer susceptibility, and then when we looked in the tumor, we knew that 90% of them were retained in the tumor. So this is important when doing tumor-only sequencing. So there are some commercial companies out there that do that, to recognize that there may be a germline variant that you're looking at.